Hi everyone, and welcome to Module 4, in which we're going to dig deeper into carbohydrate metabolism. So we've talked about how when we eat carbohydrates, they're enzymatically degraded in our digestive system, and then they're absorbed into the enterocyte and go to the hepatic portal vein and go to the liver. And then last week we talked about how they can be built up into a glycogen molecule that can help store the glucose, and then that glycogen can be degraded in order to release the glucose for energy or full body homeostasis. So this week we're going to talk about different pathways in carbohydrate metabolism. So first we're going to talk about ATP itself. What kind of properties does it have? We'll then talk about glycolysis and talk about some of the fates of some of the products of glycolysis, pyruvate and lactate. We'll then discuss the next step, which is the TCA cycle, and try to understand what is happening in some of these reactions. We're going to discuss the pentose phosphate pathway, which is an alternative pathway for glucose metabolism, and we're going to discuss gluconeogenesis. So we know that we need energy, right? We know we need food in order to produce energy. We need it to do work. We need it to build tissues and cells. We need it to maintain whole body homeostasis and cellular homeostasis. And we know we get this energy from our food, and primarily for most people from carbohydrates. So how do we get from a glucose molecule in our liver to our actual energy, which is ATP? Now, we know our primary source of energy is ATP, and that it has these high energy phosphate bonds that we can break when we need to use energy, and we can form when we have extra energy so that it can store it in those bonds. So how do we get ATP? Well, for the most part, we get most of our ATP through cellular respiration. And when we produce this ATP, again, it can serve as an energy reservoir. So here's kind of a very simplified figure of how it goes. So say we have our ATP, we break a phosphate bond and use that energy for work, and then we're left with ADP plus a phosphate, but no energy in that last bond there. And then we eat food, and the energy from the food powers the formation of this bond, so that, again, we have stored energy in the form of ATP. And we're going to see exactly how that works. Okay, so we can form ATP through two primary mechanisms, okay? The first mechanism is called substrate level phosphorylation, and this is much simpler than it sounds. All we're doing in substrate level phosphorylation is transferring a phosphate group from one molecule onto another molecule. And it can be from ATP to, for example, it could be from ATP onto glucose, which would give us a glucose 6-phosphate, right? So we took one of the phosphates from ATP and put it on glucose to get glucose 6-phosphate, like we learned about last week in glycogenesis. Or it can go the other way. We could have a molecule that has a phosphate on it that can transfer it onto an ADP, so that we have an ATP. But basically, what's going to happen is when we break, for instance, ATP, we have an exothermic reaction. It's releasing energy. And it's so highly exothermic that it makes the endothermic reaction, where the phosphate's going onto the other molecule, energetically favorable. And that's how that works. And we'll point out a lot of steps in carbohydrate metabolism where we see this substrate level phosphorylation. Now, where we get most of our ATP formation, however, is from oxidative phosphorylation. And oxidative phosphorylation, just like it sounds like, requires oxygen. And energy that we harness in the electron transport chain is used to unite ADP with an organic phosphate in order to form ATP. And we'll go into detail about how that works. Okay, so glucose can go down a lot of different pathways. So we've talked about glycogenesis and glycogenolysis, but we can also have the pathways that we're going to talk about today. So We can have pathways where we break down glucose by glycolysis and the TCA cycle, and eventually aerobically in the TCA cycle, and eventually harness that energy in the electron transport chain. We have the alternative pathway of the pentose phosphate pathway, and we also can make glucose if we have the right substrates. And the path that this glucose is going to go down is really going to depend on our cell type. So is it a muscle cell? Is it a liver cell? Is it a kidney cell? And it's also going to depend on what's going on with our body at the time. What is our physiological demand? So let's go through the different tissues and see how metabolism could vary in those tissues. Now, we know that when we're talking about glucose metabolism, the liver is central headquarters. So glucose can have a few different fates once it's come from the small intestine and it goes into the liver. So we already talked about last week, we can store it as glycogen. We can also break it down and oxidize it for energy, which is what we're going to talk about in detail today. And we can also use the carbons from the glucose in order to synthesize fatty acids or amino acids. And we'll talk about those in those pathways in those sections. So we know, for instance, if our blood glucose is low, then our liver is able to degrade glycogen to release the glucose, or if the substrates are available, we can actually make glucose via gluconeogenesis. And our hepatocyte can also, like many other tissues, but this pathway primarily happens in the hepatocyte, it can metabolize glucose via the pentose phosphate pathway, and we'll see why that's important. So now, carb metabolism is going to be a little bit different in our muscles. So again, if we have glucose in our muscles, again, we can build it up in the form of glycogen, or we can break it down and oxidize it for energy. However, we talked about last week that our muscles do not have G6 phosphatase. So our muscles are unable to convert G6P, or glucose 6-phosphate, back to glucose. Therefore, once it's in its phosphorylated form, G6P, it can't leave the cell. It's trapped in there. So our muscle cells can then use it for energy, but it can't release it back out into the system to maintain whole body glucose homeostasis. And then our needs are dependent on the type of muscle that it is. So we maybe have variable energy needs for our skeletal muscle, whereas we have a constant energy need for our heart muscle. And then in our adipose tissue, we can degrade glucose by glycolysis in order to ultimately produce glycerol that we can use for triglyceride synthesis. So if we have a really high carbohydrate intake, that can contribute to the development of triglycerides. Throughout this class, I'll use triglycerides or triacylglycerides interchangeably. They're the same thing. Also, glucose can, when it's converted to acetyl-CoA, that can be used for fatty acid synthesis. And then we have our glucose needs in our other cells as well. So we know our brain is very highly dependent on glucose in normal physiologic conditions. Under some altered conditions, it can rely on ketone bodies, but for the most part, it relies on glucose, and it completely oxidizes glucose. Now, we have other cells that don't have any mitochondria, and if they don't have any mitochondria, then they can't metabolize glucose aerobically, and therefore they metabolize the glucose anaerobically, especially to produce lactate, which we'll see. But these cells rely exclusively on glucose, but they do process it anaerobically. All right, so let's dig into metabolism. So our first pathway that we're going to talk about today is glycolysis. So as a reminder, in glycolysis, we're starting with glucose, and instead of building it up into glycogen, we're going to break it down into energy. Now, I really like this overview, and I just want to note a couple of things on this overview before we go into the details. So the glycolysis is divided into two stages. The first stage, we are using energy, and in the second stage, we are producing energy, okay? So you'll notice that in the first stage, we're using two ATPs. We use one here and we use one here. And if we look at the enzymes, they are both kinases. So not a surprise, right? These kinases are taking a phosphate group from the ATP and adding them onto the substrate. So it's going from glucose, we're adding a phosphate group. So here's our glucose here. We're adding a phosphate group from the ATP and we're phosphorylating it, and that was a kinase. So, I mean, these look like a lot of like intimidating words, but if you really start to look at them, you can start to like see what's happening. So we see that the phosphate was put on carbon number 6 here. And then, so it's glucose 6-phosphate. And then here we have fructose 6-phosphate, and again, a kinase reaction. So we're adding another phosphate onto here. So we used another ATP. And then you can see on the second part, we're producing ATP, but again, we're still using these kinase reactions. And note, there's no kinase enzymes that aren't associated with a transfer of a phosphate group. Now, one last thing that I want to point out in this graphic is this reaction here. So we only have one step in glycolysis that is going to produce these electron carriers here, okay? So one of our electron carriers is NADH. And we'll notice that in any of these reactions where we're producing electron carriers, we are reducing these electron carriers. So we go from NAD+, we're adding electrons onto it to become NADH, so that it's an electron carrier here. And what's really happening here? We're actually taking, I just want to make sure I'm saying this right. Okay, we're actually dehydrogenating our substrate and adding the hydrogen groups onto this NADH. So this is a dehydrogenase reaction, because we're moving hydrogens onto our electron carrier. Now let's look at these steps in just a little bit more detail. Okay, reaction 1 is an important one, and it's where we are transforming glucose. We have an energy input from ATP, and we're forming glucose 6-phosphate, but you can see we've used our ATP. And again, we use a kinase for this. In this case, it's a hexakinase in the muscle. It could also be a glucokinase. And what this step does is a couple of things. Number one, it phosphorylates glucose so that it's trapped in the cell. So therefore, it can't just go back out of the cell. It's trapped in the cell so that it can be used by the cell. And it also activates it. So here we just have plain old glucose, but we saw even last week for glycogenesis, and we saw on the last slide in glycolysis, that really our first step is converting glucose to G6P, because it's this G6P that can then go to all of these different metabolic pathways. So we're activating it. So we know that in the liver we have glucokinase, and that allows for kind of the it allows the liver to buffer the blood glucose, even though we don't have that ability. Sorry, that sentence is a little bit misleading, but sorry, when I first read through that, I was thinking it was G6P phosphatase, but that's not where we're going with this conversation. So basically, when blood glucose is high, the liver can preferentially trap the circulating glucose and store it, because we are phosphorylating it and trapping it in the cell. Now, glucokinase actually has a pretty low affinity for glucose, which is kind of surprising. So both the liver and kidney have glucokinase, but if we have low blood glucose levels, our glucokinase is not going to become activated, because what our body is trying to do is to spare the glucose that we have for the tissues that need it even more, like our brain. So another benefit of the glucokinase is that because it has a low affinity for glucose, that if we're starving and our body's saying, give me glucose, give me glucose, give me glucose, and we make glucose via gluconeogenesis, we're really still not going to have enough glucose in there that it's going to activate this glucokinase to transform glucose. So then it'll just, again, keep the glucose that we have to maintain our body homeostasis and to give to the tissues that really need it. Okay, and then we have reactions 2 and 3. So in reaction 2, we're just an isomerization reaction in which we're converting G6P to F6P. In reaction 3, we do have another energy using step. So we've used 1 ATP so far in glycolysis, and in this reaction 3, we're using another one. So again, it's with a kinase enzyme, phosphofructokinase, and we are converting F6P, and then we're taking a phosphate group from this ATP, and we're putting it on our fructose 6P to get fructose 1,6 biphosphate. So we've used 2 ATPs at this point, and that's the energy using stage of glycolysis. We've invested 2 ATPs at this point. Okay, and then in reaction 4, we're going to take this fructose 1,6 biphosphate, and we're going to take this 6-carbon molecule, and we're going to bust it into 2 3-carbon molecules, okay? 2 3-carbon molecules, DHAP, we'll see this again later in the course, and glyceride 3-phosphate, okay? And these are kind of going to go back and forth, back and forth, back and forth. But really what we want to end up with is this G3P, because that's what's going to make our reaction keep going. But you should know it breaks it into 2 of these, and they kind of can transform back and forth until we end up really with 2 G3Ps. So keep in mind now that we had 1 glucose molecule, but now we're down to 2 G3P molecules. Okay, so now we're entering the energy yielding phase of glycolysis, in which we're going to produce energy in electron carriers. So our next step in reaction 6 is catalyzed by a dehydrogenase enzyme. So I hear dehydrogenase, which means I think I'm going to take a hydrogen off of our substrate, our G3P, and lo and behold, here's our hydrogen, and I'm going to put it on this NAD, and I'm going to produce an electron carrier, NADH. So I think I used to get kind of confused and say, wait a minute, if I'm adding a hydrogen, isn't that positive? But notice this was positive. So really we're adding a hydrogen, which is going to add an electron as well. So that's going to be, so we're going to have an added electron here, and this is called an electron carrier. Sorry, I just want to make sure I'm not missing anything. Okay, in reaction 7, we have another kinase reaction. Sorry, the graphic here isn't very good. But in this kinase reaction, we're actually producing ATP. Phosphate of 1,3-BGP is used to form, one of the phosphates from phosphate 1,3-BGP is used to form ATP and 3-phosphoglycerate. And you can see that we have magnesium as a cofactor here. Okay, and then in our remaining steps of glycolysis, we're really trying to convert our low energy substrate into a high energy form. So in our reaction 8, we're just moving where the phosphate is in the group. In reaction 9, again, it's not one of our most important steps, but we're producing PEP. And then in reaction 10, we're converting PEP to pyruvate. And here's our PEP here, phosphoenol pyruvate. And we're going to take this phosphate group that's on this PEP and then transfer it back on to an ATP. So this is an energy producing step, and then we're left with pyruvate. And again, this is a kinase enzyme, so we know that this is going to have a phosphate transfer in this step. So again, we've produced energy, and we're left with a pyruvate. But please remember, we started with 1 glucose, we got to 2 3-GP, and then that brought us to 2 pyruvates. So we have 2 pyruvates at this point. Okay, so this figure is from your book, and it's showing glycolysis. It's in a little bit odd form. So here's our glycolysis pathway where we're starting with glucose, G6P, F6P, and ultimately down to our G3P and DHAP. So you guys remember that step from earlier. That's where we're breaking it down to these 2,3 carbon molecules. So this is just part of the glycolysis pathway here. But what this figure is showing is what happened to all the other types of monosaccharides, right? We saw that all the monosaccharides were absorbed into the hepatic portal vein, but all we've been talking about is glucose. So what happened to the other ones? Well, as this diagram shows, the other monosaccharides are also broken down and degraded, and they're broken down until they get a product that is an intermediate of the glycolytic pathway, and then they enter the glycolytic pathway just like glucose was. So you can see here with our galactose, we go through a series of reactions and ultimately end up with G6P, and then we can keep going with our reactions. We have fructose here, and we can see through a couple of different pathways that fructose can end up as an intermediate in the glycolytic pathway, and then we can just keep going with our process. And here's mannose over here as well. Okay, so we've talked about glycolysis. Now we know what happens next really depends on what our conditions are, and specifically if we have oxygen available to us. Now, if we do have oxygen available to us, then we are going to, then the pyruvate is going to be transformed into acetyl-CoA, catalyzed by pyruvate dehydrogenase, and it's going to enter into the mitochondrion. Okay, so here's our mitochondrion, here's our outer membrane, here's our inner membrane, here's our cytosol. If we have oxygen, the pyruvate is going to come into the mitochondria and be transformed to acetyl-CoA. And you can see that this reaction does produce an electron carrier. Okay, now let's talk about the TCA cycle. So we have our acetyl-CoA, we're in the mitochondria. Now we can go through oxidation, or the TCA cycle, which is our next step after glycolysis if we have an aerobic environment. So here, one thing I want to point out, we'll go through all of these steps, but I just kind of want to do a little overview here. So we have acetyl-CoA, but acetyl-CoA is not powering this whole cycle itself. Instead, it needs to join up with oxaloacetate, and those two combined can then keep this cycle going. And we can see that we have a lot of products in this TCA cycle. So we produce NADPH, we'll talk about that. We'll produce carbon dioxide, we'll produce NADH, we'll produce ATP, we'll produce FADH. So we have a lot of products of this. And our ATP, we know, is our direct energy, and our FADH2 and our NADH are our electron carriers that will ultimately produce energy after they go to the electron transport chain. Okay, so first, when we're talking about the TCA cycle, some of our most important reactions where we're producing energy or producing electron carriers that will ultimately end up in producing energy, I should say, are our dehydrogenase reactions. I'm going to go back a little bit. Okay, so you can see here that when we produce NADH, when we produce FADH2, produce NADH, these are all these enzymes are called dehydrogenases. Dehydrogenases. So let's go to the next slide and see what's going on here. Okay, so these represent two different steps of the electron transport chain. So in this one, we're converting malate to oxaloacetate. So we can see here that malate dehydrogenase. So we're going to take some hydrogen off of malate, and we're going to put it on this NAD+. And in that way, not only are we donating hydrogen, we're also donating an electron onto this NADH, hence why it's called an electron carrier. So a dehydrogenase enzyme is going to take hydrogen from our substrate and put it on this electron carrier. And then we're left in this stage with oxaloacetate, which we can see has fewer hydrogens. And then this NADH is then going to go to the electron transport chain. Here's another step of the TCA cycle where succinate is transformed to fumarate. And again, we have a dehydrogenase reaction, which means we're taking the hydrogen off of the succinate. And in this case, we're adding it onto FAD+. In therefore doing, we are so we're oxidizing succinate, right? Because we're removing hydrogens. We're oxidizing succinate into fumarate, and we're reducing FAD into FADH2. And so this is now an electron carrier, and that's going to go to the electron transport chain. All right, so let's go through the TCA cycle in detail. And I did put a little asterisk by some of our more important steps. So first we have, our first step is to produce citrate from the combination of acetyl-CoA and oxaloacetate. Okay, and then in reaction 2, we're just creating an isomer of citrate, which is isocitrate. So that's not our most important reaction. Okay, and then in reactions 3 and 4, we're going to produce electron carriers. So these are really important steps. So in reaction 3, we are transforming isocitrate to alpha ketoglutarate. And again, we can see that some of our hydrogens from our isocitrate are going to reduce NAD+, so that it becomes NADH2. That's going to go to the electron transport chain. And we can see it was isocitrate dehydrogenase, so it's a dehydrogenase enzyme again. And same thing for reaction 4. We have a dehydrogenase enzyme, alpha ketoglutarate dehydrogenase to be exact. And in this reaction, we are again going to oxidize alpha ketoglutarate to succinyl-CoA while we reduce NAD+ to NADH. And then the NADH goes to the electron transport chain. And you can see that we also have CO2 as an output here. Okay, in reaction 5, this is actually the only step in the TCA cycle that is going to give us direct energy. And in some figures, you might see it as ATP, and some you might see it as GTP. It doesn't really matter. They both have the high energy phosphate bonds. You can really look at this as an ATP if you see it as a GTP, because what's going to happen really is that an ADP is going to come in here and become an ATP, and then GTP will go back to GDP. So this is a step where we get direct energy via substrate level phosphorylation. Okay, in reaction 6, we have another dehydrogenase, which is a signal to us that we are going to reduce FAD to FADH2. That's going to go to the electron transport chain. Reaction 7 is a hydration reaction, not one of our more important ones. Okay, and then in reaction 8, we have another dehydrogenase reaction, and again, we are going to transfer our hydrogens onto our NAD, so we produce NADH, electron transport chain. Okay, so what is our overall equation? We are going to start with acetyl-CoA from glycolysis. We need to add in NAD+. Keep that in mind that we need this in order to make the TCA cycle go. We're going to need FAD, we're going to need GDP, plus an inorganic phosphate and some water. And we're going to produce carbon dioxide. We're going to produce 3 NADHs, because we saw it in every different step of the cell where we produce those NADHs. We're going to produce 1 FADH2, 1 GTP, and then some hydrogen in our co-wash. So that's our overall equation for the TCA cycle. Now, one thing that you need to remember is that the TCA cycle turns 2 times for every glucose, because every glucose is going to produce 2 pyruvate, okay? So everything will look like it's doubled, okay? So when we're adding up how much energy we're going to get. So let's look at how much energy we add up. Now, first, think back to your biochemistry class. Now, we know that NADH is going to ultimately help us produce 3 ATPs. And FADH2 is going to ultimately help us produce 2 ATPs. These other numbers in parentheses you might see in your readings. Sometimes we say it's worth 2.5 ATPs. But just for simplicity's sake here, we're going to say that each NADH will help us produce 3 ATPs. We'll see how that works. But for right now, let's just look at where we're getting all of this energy from in glycolysis and the TCA cycle. So we know that in glycolysis, we produced 2 NADHs, so 2 times 3 ATPs, that ends up with 6 ATP equivalents. And then we have 2 cases of substrate level phosphorylation, where we're going to get 2 each from there. So in glycolysis, we produced 10 ATP, but remember, we used 2 in the activation phase. So therefore, our glycolysis gives us a net of 8 ATPs. And then we saw that in 3 different stages of the TCA cycle, we produced an NADH. But remember, for every glucose, we're going to spin the TCA cycle twice because we have 2 pyruvase. So each time we get 2 NADHs, really, 1 for each spin of it. And then we end up with 6 for each step, if that makes sense. So we saw we produced 1 NADH, but we actually have 2 pyruvase, so I'll produce 2 NADHs, and each NADH is worth 3 ATP. So we see there, there, there. And then we have that production of GTP/ATP. So we had a substrate level phosphorylation there, times 2 turns is 2 ATP. And then we had our FADH2, and then, we have one more NADH here. So in the end, from our TCA cycle, we get 30 ATP. Net, we have 38 when we combine glycolysis and the TCA cycle, but we use 2. So we end up with 36 total. So you can see that that initial investment of 2 ATP in glycolysis really was worth it, because we ended up with 36 more ATP than that. So if you want to review the Krebs cycle, I'm going to let you go on your own to this link that will review all the steps of the Krebs cycle. And glycolysis. All right, so, sorry, sometimes I get mixed up what video we're on. Okay, yes, we are going to watch this one. So this is a review of the Krebs cycle in a fun song way.
Matrix of the mitochondria of all our cells is a cycle of reactions that one hands Krebs a Nobel Prize. The cycle takes the energy and food and makes it into other forms your cells can use. Krebs cycle makes electron carrier NADH, which later brings electrons to the electron transport chain. And Krebs makes FADH2, its function is the same. Krebs also makes some ATP, another claim to fame. We're talking Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Right before the cycle's a transitional part, links Krebs to glycolysis so Krebs can start. Enzymes break the CO2 off a pyruvate, yields an NADH and acetyl-CoA. Acetyl-CoA carries carbons too, with all the energy that derives from food. It's a highly reduced molecule with energized electrons that fuels up the Krebs cycle as it cycles on. Now after start of Krebs, this acetyl-CoA has the 2 carbons it carries in with the weight. Enzymes hook these on oxaloacetate with carbons 4, makes 6 carbon citric acid. Who could ask for more? Notice 3 carboxyl groups on the citric acid. That's why the cycle's also named for tricarboxylic acid, or TCA cycle. It's acronyms that you prefer. Krebs in honor of its discoverer, Krebs. It's a citric acid cycle, Krebs. Tricarboxylic acid cycle, Krebs. Each cycle makes 1 ATP, 3 NADH, 1 FADH2. Enzymes work on citric acid and remove the CO2, and other enzymes modify and oxidize it too. The 5-carbon result is alpha-ketoglutarate. Krebs cycle, it's so great. Every oxidation can power the reduction of NAD+, which gains electron carrying function, become an NADH, that energy sensation, which later on in respiration powers ATP creation. Another oxidation follows another CO2 removed, leaving us with a 4-carbon molecule. Another NADH results from this oxidation. Krebs does its energy transformations. This 4-carbon molecule, succinyl-CoA, still has lots of energy enzymes can take away. A series of reactions yield 1 1 ATP, the cell's main energy currency, leaving just enough energy for enzymes to reduce NFADH to FADH2, and one less NAD plus will also get reduced as the final electron carrier NADH gets produced. We've harvested what energy came in at Krebs to start. Now we have oxaloacetate at the final part. Oxaloacetate is the commencement and finale, ready to meet acetyl-CoA and here at the final tally. Krebs goes round and round, such an ancient cycle, spinning like the wheels of my bicycle. Krebs like the axle of aerobic respiration. I breathe out CO2 with every exhalation. Walk through the cycle, so now let's review. Input is acetyl-CoA with carbon 2. The carbons get removed, releasing CO2. Exhaling sends the CO2 out of you. The cycle's functions: energy transformation, 3 NAD, H1 FADH2 creation, and also synthesis of 1 ATP, which cells directly utilize for energy. For every glucose cells absorbed, the cycle runs 2 times. As long as cells get fuel, Krebs is running just fine. It proceeds electron transport chain, it follows glycolysis, and spinning round and round in the mitochondrial matrix. Krebs, it's a citric acid cycle. Krebs, tricarboxylic acid cycle. Krebs, the cycle makes 1 ATP, 3 NADH, 1 FADH2. Krebs, it's a citric acid cycle. Krebs, tricarboxylic acid cycle. Krebs, the cycle makes 1.
Okay, I really like that video, and I like how they track the different numbers of carbons as they go throughout. So I did make a minor mistake before we went to the video. So the TCA cycle does only produce 3 NADH, 1, 2, 3 per turn, but we also get an NADH from the pyruvate dehydrogenase reaction, where we're transforming pyruvate into acetyl-CoA. So just to clear that up. Okay, so how do we regulate TCA cycle? Well, we have 2 major messengers that provide feedback for how the rate that we're going to run this TCA cycle. And they're 2 of our big products. So the first one is pretty intuitive. It's ATP, the phosphorylation state of ATP. So our body is able to measure the concentration of ATP, which is our high energy state, with ADP, which is our lower energy state. So basically, if I have a lot of ATP in my cell, then what's the rush, right? We don't need to run the TCA cycle like crazy if we've already got enough ATP. So ATP is actually going to inhibit the TCA cycle from running. Whereas ADP, if we've got a lot of ADP, then our body's going to say, okay, or our cell is going to say, I need some energy here, folks. Let's run this TCA cycle double time. Let's get some energy. So that's one of our messengers that's going to provide feedback. And our other messenger is our concentration of NADH to NAD+. So our reduced NAD to our oxidized NAD. And it's the exact same thing as with the ATP. So if one of our products is NADH, and we have a lot of NADH in our cell, then our cell is going to say, okay, don't make any more NADH. I've got to take care of this NADH I've got. Let's slow down the cell, or slow down the TCA cycle. And I like this graphic here. I think it shows it really nicely. But let's look at a couple of places where, so for instance, how about right at this step with the citrate synthase, we can see that if we have ATP or NADH, those are going to inhibit this enzyme from making the cycle go, where we're going to produce more ATP and more NADH. Counter to that, though, if we have a high ADP, then our cell's going to say, oh, let's keep running this thing, let's make some energy, and it's going to stimulate this enzyme. And just like any of our other biological processes, our bodies are very adept at sensing quickly changing concentrations and reacting to them in order to maintain homeostasis. Okay, now let's take a little step back and look at a different part of this. Now, we saw all of our intermediates in our glycolytic pathway and in our TCA cycle. And actually, these intermediates are not only used in the TCA cycle, but they can also be taken out of the TCA cycle in order to synthesize other molecules or sorry, I guess you could say molecules or structures. So for instance, citrate can be taken out of the TCA cycle in order to start to biosynthesize fatty acids. Oxaloacetate and alpha-ketoglutarate can be used to produce amino acids. And when we remove these intermediates from the TCA cycle, well, then it's a cycle, it's a circle. It's going to stop the cycle if we remove the intermediate. So if we do remove the intermediate to make something else in the body, then we need to be able to replenish it as well. All right, so let's talk about an alternative pathway of glucose metabolism. So we talked about how it's broken down through glycolysis, and if we have enough oxygen, it can go through the TCA cycle. Now, we also have a different pathway, and that's called the pentose phosphate pathway, also called the hexamannose phosphate shunt, but I'm going to keep it as pentose phosphate pathway just to keep us all on the same page. So, in this pentose phosphate pathway, we can produce two substances that are really important in our bodies. So the first one that we produce is NADPH, and we use NADPH for fatty acid and steroid biosynthesis, and we'll talk more about that in the lipid section. And it also helps to the red blood cells to maintain functional integrity and membrane integrity, and we also use NADPH for drug metabolism. Now, in addition to NADPH, the pentose phosphate pathway also helps us produce the 5-carbon sugar ribose 5-phosphate. And that should sound pretty familiar because that is a substance that we need to produce nucleotides and nucleic acids in DNA and RNA, of course. Okay, so the pentose phosphate pathway can occur in many different types of tissues, especially in our liver. It operates in the cytoplasm. But in other tissues, such as the skeletal muscles, it actually has really low activity. And that kind of makes sense because in our skeletal muscles, we have less of a demand for things like fatty acid synthesis and therefore less of a demand for NADPH, which will help us produce fatty acids. And instead, in our skeletal muscle cells, we're relying on glucose and fatty acids for energy. We're not trying to synthesize fatty acids necessarily. All right, so let's look at the the pentose phosphate pathway. So, okay, let me see if I can get this one straight. So, it has two different phases, the oxidative phase and the non-oxidative phase. So this is kind of like two parts to the same picture. This has the oxidative phase in detail, and this has the non-oxidative phase in detail. So in the oxidative phase, our G6P, hopefully that looks familiar, is our main substrate, and it's going to generate a mole of NADPH, which is catalyzed by the G6P dehydrogenase and the 6-phosphogluconate dehydrogenase. So all you need to know is they're dehydrogenase enzymes, and those are going to help produce NADPH. And I don't know if from your prior classes that you recognize this glutathione reductase, but that is utilized by our red blood cells. Okay, and now, we keep going through this pathway, and we ultimately produce not only NADPH, but we also can produce ribose 5-phosphate. That's going to be integral part of our nucleotides and our DNA and our RNA, for instance. So that was our oxidative phase. And then we have our non-oxidative phase in which ribose 5-phosphate can actually be transformed back into glycolytic intermediates, such as fructose 6-phosphate and G3P. Here's fructose 6-phosphate again, and G3P, and these can go back to G6P. And we know that G6P is kind of the start of our metabolic pathways. other metabolic pathways. Okay, so we talked a lot about what happens if we have oxygen. Then the pyruvate can go to the acetyl-CoA and the mitochondria, and we can go through oxidative phosphorylation. But what about if we don't have enough oxygen? What if we're in like a red blood cell that doesn't even have a mitochondria? What are we going to do? Well, at that point, instead of pyruvate being transformed to acetyl-CoA, instead it's going to be converted to lactate with the lactate dehydrogenase enzyme. And this step is important. So we will see that we get way more ATP from aerobic glycolysis compared to anaerobic glycolysis. But anaerobic glycolysis is important because, number one, it helps us break down glucose even if we don't have oxygen, but also it helps us produce this NAD+. And if you'll remember from our earlier in our discussion, we need this NAD+ in order to keep the TCA cycle going. So it's one of our inputs that we have to have, and we can get that from the lactate metabolism. So once we do have lactate, so once pyruvate is transformed to lactate, it can actually be oxidized back to pyruvate, and then if we have oxygen at that point, then it can go into the TCA cycle. And if energy levels are high, that pyruvate can actually undergo gluconeogenesis and go back to glucose. So let's break that down for a minute. If energy levels are high, if I have a ton of ATP, then pyruvate's not going to need to break down and make me more ATP, right? We can go back up to gluconeogenesis and put it into glucose form. Okay, so here's a little graphic. So here, think we're in glycolysis. We have G3P, we have 1,3-bis-P glycerate, and then we have pyruvate. And if we do not have oxygen, then this pyruvate is going to, remember this is called lactate dehydrogenase, right? So we're going to take the hydrogen off of NADH, and we're going to produce this NAD+ that we need for the TCA cycle, and pyruvate is transformed to lactate. And then here's just kind of a nice overall picture that's showing you what's happening. So here we have our glucose, and then our G6P, which can then go to glycogen metabolism, or we can break it down by glycolysis to pyruvate, and then pyruvate can either, in aerobic conditions, go to acetyl-CoA and then to the TCA cycle, or pyruvate without oxygen can go to lactate via lactate dehydrogenase. Okay, so we know this just as a little review. Once acetyl-CoA is completely oxidized in the TCA cycle, we produce CO2, we produce direct energy in the form of GTP or ATP, and we produce reduced electron carriers. That's our NADH and our FADH2. And then those reduced electron carriers go to the electron transport chain. And it's important to know that this cycle does have several required cofactors, such as thiamine and riboflavin. All right, so what is oxidative so we've come to oxidative phosphorylation. So we saw it in our TCA cycle and in our glycolysis and then TCA cycle that we had our dehydrogenase enzymes, and the dehydrogenase enzymes transferred hydrogens, which came with an electron, right? It transferred them from a substrate onto this either NAD or FAD, and so now these become electron carriers, okay? And then these electron carriers are going to go to the electron transport chain. And this is where we really have our oxidative phosphorylation to produce ATP. So here's what's going to happen. The electron carriers are going to go to the electron transport chain. And what the electron transport chain is a series of intermediate compounds which are going to pass the electrons through them in a downhill reaction and ultimately pass these electrons onto oxygen to produce water. If this is confusing, don't worry, I've got some figures coming up. So, and it's this, Okay, so when I say that they pass electrons from compounds to the other, from one compound to the other, these are redox reactions. So remember to your biochemistry, and we've been talking about redox reactions throughout this whole talk, right? So if I have NAD and I add a hydrogen onto it, then this is going to go from being the oxidized form, the NAD, to the reduced form, NADH. That's a redox reaction. And whatever molecule passed the hydrogens onto NADH, it went from being reduced to being oxidized when it passed these hydrogens onto the NADH. So in order to reduce something, we can either take away an oxygen, we can add electrons, or we can add hydrogens onto it. And oxidation is the same. But basically, when you have two molecules in a redox reaction, one of them is going to be oxidized and one of them is going to be reduced. So if I'm passing hydrogens from one molecule to another, the first molecule is going to be oxidized, the second molecule is going to be reduced. So, so when the electrons go through this series of carriers on the electron transport chain, it's going to create an energy gradient. And we'll talk about why. And that energy gradient is going to power the phosphorylation from ADP to ATP. That's what oxidative phosphorylation is. Okay, so just a little reminder of our biology. Here we have our mitochondria, we have our outer membrane, we have our inner membrane, and we have our intermembrane space between the inner and outer membranes, the inner and outer membranes. And our electron transport chain is located in this inner mitochondrial membrane, and all of these complexes are set up in a row in this membrane in order to facilitate the uptakes of electrons from our electron carriers. And let's see. Okay, and then each component of the electron transport chain is going to first accept the electrons from the electron carrier and then from the other components, and then it's going to donate the electrons as it moves down the chain. And so as these electrons move down this chain, it's going to release energy. And this energy is going to power, again, I'm going to go through all of this again. This is just introduction. This energy from the electrons passing down these complexes in the electron transport chain are going to power a translocation of hydrogen ions from the interior of the mitochondria into the intermembrane space, and it's going to create a concentration gradient. And this concentration gradient is what we're going to use to make our ATP. Okay, so here we have our four main parts, complex 1, 2, 3, 4, of our electron transport chain. Okay, let's start with complex 1. So we have our electron carrier here, NADH, okay? The electron from the H is going to go on to complex 1, okay? The hydrogen is going to go into the mitochondria. This is the inner mitochondrial membrane, remember? Okay, so this electron is then going to get passed down to complex 2, then to complex 3, then to cytochrome 3, then to complex 4. And this passing down of electrons is kind of a downhill reaction. And what that downhill reaction is going to do is power these hydrogen ions in the membrane and power them through these different parts of the electron transport chain and make this concentration gradient of heavy hydrogen ions in the intermembrane space. These are then going to be attracted to this other side of the membrane, right? They can't diffuse through the membrane, so they're going to be attracted to this other side of the membrane because they're in such high concentration on this side. And when they go back through, so they can get back into the membrane through this ATP synthase, and the hydrogens falling down their concentration gradient is what's going to power this transformation of ADP to ATP. Okay, so we have 4 different complex structures in this electron transport chain on the inner mitochondrial membrane. So, so in complex 1, we're going to have our NADH reaction occur, and we're going to have NADH dehydrogenase. Makes sense. Going to take the hydrogen off of the NAD, and the hydrogen is going to be in our mitochondrial matrix, and we're going to pump our electron to complex 2. In complex 2, this is where our FA, it doesn't say that here, I wish it would, but we're going to have, this is where our FADH2 is going to come in, and again, we have a dehydrogenase, and the hydrogens are going to be in the mitochondrial matrix, and the electrons are going to get passed down to CoQ10. Now, as the electrons are getting passed down, these redox reactions are actually occurring in these complexes as well, and it's creating energy, and the energy is pumping these hydrogens again into this intermembrane space, And so complex 3 is going to carry electrons from CoQ to cytochrome C, and then the electrons are transformed onto complex 4. And then this is kind of the final destination for these electrons, because these electrons are then going to reduce oxygen to water. Okay, so how does this work? So how can we pass electrons from one complex of the electron transport chain to the other? Well, basically, complex 1 and 2 in the CoQ system all have these iron-sulfur centers. And if you remember back to your biochemistry, we know that iron can be transformed from its 2+ to its 3+ version. So the electrons can be, so it can change its state when the electrons are passed onto it. It would become a Fe2+, and then it would pass the electron on to the next complex in the system, and it would become Fe3+, for instance, as it reduces the next complex. Okay, and so I think that's all I need to cover on this one. Okay, so at this point, we have all of our electrons have passed down our units in our electron transport chain. We finally reduced oxygen to water, and we know all of this has powered the pumping of hydrogen ions against their gradient into the intramembrane space. The hydrogens can't get back into the space, and so they've got to find a way to get back inside. And the way that they can get back inside is in this ATP synthase enzyme complex. It's just like a lot of different, which has a lot of different parts to it. So the hydrogens want to come back into the, hydrogen ions want to come back into the mitochondrial matrix. So they find a way, they go back through this ATP synthase, and this energy that we get from them falling down their concentration gradient is actually what's going to cause this ATP synthase to produce ATP. And then I'm just going to if you want to watch this one on your own, I'll let you do that on your own, because it's just like a little bit longer review of everything we've talked about with the TCA cycle up until this point. Okay, so the influx of protons through, or hydrogen ions, back through this ATP synthase causes the you could think of it as like a rotor turning for this big enzyme complex. And what it does is it catalyzes the conversion of inorganic phosphate in ADP so that it becomes ATP. So here's a really short little video that demonstrates that. I think it is.
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
Okay, so this is just one more example of how amazingly complex our body is in its ability to regulate our biological processes. So this is our main method of ATP or energy production, where we have glucose, it goes through glycolysis, it has enough oxygen that it can go into the TCA cycle, and then those and electron carriers go to the electron transport chain, and then we produce ATP. So again, it's very complex, but then when you look at each step, the mechanisms are pretty consistent. We're transferring hydrogens, we're transferring electrons, we're transferring phosphate groups. There really is a language to how all of this works. Okay, so this whole so now this slide is about chemiosmotic theory. We've already explained chemiosmotic theory. Basically what it is that energy from the return flow of protons furnishes energy for the ATP synthase to produce ATP. The whole thing is called chemiosmotic theory. Okay. And here is a figure from your book, complete oxidation of pyruvate in the mitochondria via pyruvate dehydrogenase complex, the citric acid cycle, and electron transport chain. So here again, this is an overview of everything we've seen. Glucose, glycolysis to pyruvate, this should be our mitochondria here, pyruvate to acetyl-CoA, acetyl-CoA to TCA cycle, electron carriers to, or reduced electron carriers to the electron transport chain, oxidative phosphorylation, ATP. Easy as that. So let's switch gears here. Don't worry, this won't go on forever, but let's switch gears here. So we've talked about how we break down glucose. But what about when we need glucose? Okay, so we know we can degrade glycogen and we can release glucose from glycogen. Great. Well, there's another way we can get glucose. So we talked about break down glycogen to get glucose, but we can also take substrates and build up to get glucose. And this is a system, a kind of a backup system that our body has in order to provide our body with the necessary glucose that it needs for fuel. So this is especially important in our red blood cells, brain. It's important in the kidney and in the testes as well. Because again, these tissues are the sole, rely on glucose as the sole energy source, except for the brain in some situations can rely on ketone bodies, can derive energy from ketone bodies. And gluconeogenesis, do not get too scared because it's basically the reverse of what we just talked about in glycolysis. All right, so again, don't get scared. So here was our glycolysis steps where we're breaking down glucose all the way to pyruvate. Well, in gluconeogenesis, almost everything is the same. You can see the a lot of the enzymes are even the same, but we're just moving in the other direction. So we're taking phosphate or pyruvate as our main substrate, and we are then powering the reaction in the opposite way so that we can produce glucose. And one thing to note here is that pyruvate does need to combine with oxaloacetate to get back to PEP. And so here it's kind of just showing how these two processes are the opposite of each other. But here's just a little bit more detailed view of gluconeogenesis and our energy using reactions. So you can see here, so we're starting with pyruvate, and we're trying to get back to glucose, but we can see this is using energy. We are using energy in this step, we're using energy in this step, we're using energy in this step. So this is an energetically expensive process. So in order to get 2 moles of pyruvate, because remember it takes 2 moles of pyruvate to go back to glucose. This is going to cost us actually 6 ATPs. So it's very costly energetically, considering that in glycolysis, to get from glucose to pyruvate, we only used 2 ATPs. Okay, so again, we've moved backwards. So we've gone from pyruvate to, let's see, this says 1,3-bisphosphoglycerate. So where's that one? 1,3-bisphosphoglycerate. So again, we've used 2 or 6 ATP just in these first few steps of gluconeogenesis. Okay, and in the kidney and in the liver, we talked about, when we were talking about glycogenesis, that once we transform, in the liver especially, a lot of the glucose that it gets for incorporating into glycogen comes from gluconeogenesis, rather than all of it coming right from the diet. But, of course, we know that skeletal muscle lacks G6 phosphatase, so we can't really, so we might be able to get ourselves back to the G6P spot with the gluconeogenesis, but we can't really do full gluconeogenesis in the skeletal muscle cell because we can't get it back to that glycogen because we don't have the glucose 6 phosphatase. So really, if we are going to go through gluconeogenesis in the muscle cell, the only real fate of that is that it can be then stored as glycogen. It's not going to be released to the rest of the body. All right, so what do we need in order to make glucose? What are our substrates for making glucose? Well, we've got a few of them. So first we have lactate. And where do we get lactate? From pyruvate, right? We make lactate when we have anaerobic conditions once we have pyruvate. So pyruvate goes to lactate in anaerobic conditions. So this is actually the predominant source of carbon atoms that can be used for gluconeogenesis. We talked about where lactate came from, and especially it comes from our cells that don't have mitochondria, like our red blood cells. And the lactate is released into the bloodstream and then goes back to the liver to be used as a substrate for gluconeogenesis. And this cycle is called the Cori cycle. So basically what it does in the Cori cycle is it utilizes glucose from other non-hepatic tissues and uses that carbon source for gluconeogenesis. And here's our Cori cycle. So we're in the muscle, we are, through glycolysis, we produce 2-pyruvate, but we don't have enough oxygen, so we produce lactate. The lactate goes into the blood, to the liver, at which point we go through gluconeogenesis to get, at which point the lactate is converted to pyruvate, and then we can go through gluconeogenesis to produce glucose in the liver, and then that glucose can go to the rest of the body. So though we can't produce glucose from gluconeogenesis for glucose in the rest of the body, we can use the lactate from the muscle, bring it to the liver, and then produce the glucose for the rest of the body. Okay, beyond lactate, of course, we can use pyruvate for gluconeogenesis. And this is generated in muscle and other peripheral tissues from glycolysis. Pyruvate is also an important substrate because it can be transaminated to alanine. We'll talk more about transamination in our protein section, and then return to the liver for gluconeogenesis. So if we'll remember back again to our biochemistry, what transamination is it requires an alpha amino acid to donate an amino group, and then that creates an alpha keto acid. And we use that alpha keto acid for gluconeogenesis. Sorry, let me make sure I'm getting this right. So in this glucose-alanine cycle, we have pyruvate in the muscle that can be transaminated to alanine, and then that alanine can go into the bloodstream to the liver, at which case the amino group is taken off of the alanine and it's converted back into pyruvate, and then pyruvate can be used for gluconeogenesis in the liver, or it can be oxidized in the TCA cycle. And then the nitrogen that was taken off can be converted to urea. And again, we're going to talk a lot more about this process in the protein section. So the major function of the glucose-alanine cycle is it allows non-hepatic tissue to deliver an amino portion of a catabolized amino acid to the liver for where it can be used for gluconeogenesis, and the amino portion can be excreted as urea. So let's look at this. So we have glucose in the liver, the glucose goes to the muscle, we have glycolysis, we have pyruvate from glycolysis, we add an amino group and convert it to alanine. The alanine can go into the blood, back to the liver, it's deaminated back to pyruvate that can be used for gluconeogenesis, and then the amino part can be excreted in the urea. Okay, and then here's the Cori cycle and glucose-alanine cycle together. So let's look at these. So let's look at the Cori cycle first. So glycogen, G6P, the glucose goes out of the liver, it goes to the muscle, it's degraded to pyruvate by glycolysis, not enough oxygen will become lactate, and it goes back to the liver, and then our whole cycle starts again, okay? So that's the Cori cycle. However, the glucose can then be degraded to pyruvate in the muscle, or it can, and then it can be transaminated to alanine, and the alanine can then go back to the liver to be deaminated back into pyruvate for gluconeogenesis. Okay, another substrate for gluconeogenesis is amino acids. So almost all of our amino acids, except leucine and lysine, can be degraded so that they produce TCA cycle intermediates. And those intermediates can then enter into the TCA cycle and kind of keep the system going. So if you think back to earlier in the lecture when we talked about that we have to take intermediates out sometimes, we also can make intermediates to put in sometimes. So in this way, it allows the carbon skeletons of amino acids to ultimately be used for gluconeogenesis. So we can use it to make glucose when we really need it. So basically how this all plays out is that when glycogen stores are completely depleted, we really need to make more glucose, then our body can actually catabolize our muscle protein and then use that as a carbon source for blood glucose maintenance. And I should say also that amino acids that can be used for gluconeogenesis are called glucogenic. Okay, we can also use glycerol for a substrate for gluconeogenesis. So again, part of this is going to make a lot more sense when we're in our lipids unit. But basically, when, I'm sure you remember the term beta oxidation. So fatty acids, when they're degraded and they break down, they go through a process called beta oxidation, and it produces acetyl-CoA. So obviously we know that acetyl-CoA can be used as our first substrate in the TCA cycle. So when beta oxidation breaks it down to fatty acids down to acetyl-CoA, then that can enter the TCA cycle. And then during the TCA cycle, however, we saw in that musical video that when we go through the TCA cycle, we're losing 2 carbons. So what? From our acetyl-CoA, that was only a 2-carbon unit in the first place. And if we're losing 2 carbons in our TCA cycle, well, then we don't really have a net conversion of fatty acids to carbohydrate. However, when we break down lipids, the glycerol backbone of the lipid can be used for gluconeogenesis. So if you'll remember, one of our steps of glycolysis was G3P, or glycerol 3-phosphate. So really, we could phosphorylate the glycerol backbone of a lipid by glycerol kinase and produce one of our intermediates of glycolysis that then we could use for gluconeogenesis. Okay, and then I believe this is our last substrate for gluconeogenesis, can be propionate. And propionyl-CoA is a product of the degradation of fatty acids that have an odd number of carbon atoms. And of some amino acids. We'll talk more about that in the other sections, but just know that propional-CoA can be converted to succinyl-CoA, and that's obviously a TCA cycle intermediate. So we have propionate, the glycerol backbone of lipids, amino acid, carbon skeleton, and pyruvate, and lactate. All of those can be used for gluconeogenesis to make new glucose. Now, how do we regulate glycolysis and gluconeogenesis? So remember, glycolysis, we're breaking the glucose down. Gluconeogenesis, we're building a substrate back up into a glucose. And no surprise that these have a lot of different ways that they're regulated, depending on what our needs are. So we have hormonal regulation by our friends insulin and glucagon. We have regulation by catecholamines and glucocorticoids. We have regulation by phosphorylation of our enzymes. We have coordination between some of our enzymes, especially in our liver. And we can also regulate enzymes through gene expression, through allosteric modification, and through feedback mechanisms. So again, our body has a lot of checks and balances to help us maintain this full body homeostasis when we're talking about glycolysis and gluconeogenesis. So one more point to that. So let's talk about this one about the coordination between the glucokinase and the G6 phosphatase in the liver. So basically, if there's so if you think about it, these two are kind of counteractive to each other. So the glucokinase is going to add a phosphate group onto the glucose, and the G6 phosphatase is going to take it off. So basically, we want some coordination between these two so that it limits this kind of futile cycling back and forth. We want these to be hormonally regulated so that we're not just going back and forth and using up energy for no reason. Okay, and so this is a very intense graphic from your book. Don't get too intimidated. Let's walk our way through it a little bit. So these are major points of regulation of glycolysis and gluconeogenesis in the liver. So if you go through these, a lot of them are exactly what we have talked about before. So I'll only walk through some of it with you so that you don't get bored to death, and that so you can figure some of this out for yourself. But let's just look at some of these up here. So glycolysis. Okay, so glycolysis is where we're breaking down glucose. Okay, so we see that fructose 6P is going to have a negative impact on this happening. Well, that kind of makes sense, because if we have fructose 6P here, right, it's saying, okay, we've already got a lot of this product. Why do we need to keep breaking this down so we have more product? That doesn't make sense. So okay, that's pretty logical. What about insulin? Insulin. So insulin is telling my body that I have plenty of glucose. So insulin's going to tell my body to break down the glucose to make energy. So if I have a lot of glucose, I want to get rid of it. I can either store it as glycogen or I can break it down via glycolysis. So that makes sense. Insulin's saying you have too much glucose, break it down. Makes sense, okay? And then on the other hand, here we have that insulin is going to have a negative effect on this G6 phosphatase. So why would that be? So if I have plenty of glucose, well, I don't need to make more glucose via gluconeogenesis, so it makes sense that enzyme is going to be, or that insulin is going to be an inhibitor of gluconeogenesis. So a lot of this makes a lot of sense as you go through these steps. So, and a lot of them is redundant with what we've seen. So we see here that ATP is a negative, is an inhibitor of the 6-phosphofructokinase. Well, that makes sense, right? Glycolysis is to make energy. If I've got plenty of energy, I can inhibit this whole process instead of making it keep going and going. So if you walk through these, a lot of them just kind of make intuitive sense, even though it looks really complicated. Okay, we've come to the end. So I am gonna have you do these on your own and think about the processes of these, and if you understand what these questions are saying. Of course, I am available for questions, as is Chelsea. So I look forward to reading your discussions if you are in the online class, and otherwise we will discuss them in person if you are in the in-person class or on campus class. And I will look forward to talking to you all next week. Thanks and have a good day. Bye.